Hello everyone, welcome to ISF College of Pharmacy Dialogue Series. As you know that ISF College of Pharmacy Moga Punjab is conducting a dialogue series in which online eminent pharmaceutical scientists from pan India are coming to this platform and delivering their talks. In this, today we have very special guest with us, Professor Dr. Pradeep Golani, Professor of Pharmaceutical Chemistry, Gyan Ganga Institute of Pharmacy, Jabalpur, Madhya Pradesh. Without taking much time, I would like to introduce you to our audience, sir. Dr. Pradeep Golani is an esteemed academician, researcher in field of pharmaceutical chemistry, currently serving as the head of the department at Gyan Ganga Institute of Technology and Sciences, Pharmacy Department in the state of Madhya Pradesh, Jabalpur. If we talk about his education and academic background, he completed his master's from reputed SRM University, Chennai, where he demonstrated exceptional academic excellence. If we talk about research and publications, as a prolific researcher, Dr. Golani has published 25 papers in reputed national and international journals. His research contributions have significantly enriched the field of pharmaceutical chemistry, providing valuable insights into various aspects of drug development and discovery. Dr. Golani expertise in field of pharmaceutical chemistry in terms of book writing. He is a good author. Approximately have written 7 books of the subject. His books have become valuable resources for students and professionals alike, contributing to the dissemination of knowledge in the pharmaceutical domain. Furthermore, Dr. Golani innovative research has led to the grant of one international patent on anti-cancer drugs, a testament to his groundbreaking work in drug design and development. Additionally, 6 patents are currently under process, showcasing his ongoing dedication to finding novel solutions in the pharmaceutical field. He has been member of various professional societies like APTI, ISPER, IPA, IPG, etc. He has attended various conferences. He has got various prestigious awards to his research acumen. He has got various Best Pharma Research Award into various summit. And if we talk about his academic journey, he is a pharmaceutical educator, researcher, and has been marked by numerous achievements and locates. His extensive research publications, authorship of books, patent grants showcase his significant contribution in the field of pharmaceutical chemistry. So, thank you, sir, and welcome to ISF College of Pharmacy. Now, I would like to ask you to unmute your mic and, sir, start your presentation.
Thank you so much, sir, for introducing me very specially. And I have got this opportunity to present a lecture in your lecture series. It's a good, very big opportunity for me. And I would like to thank the chairman, sir, vice chairman, sir, GD Gupta, sir, Narang, sir, and chairman, sir, Praveen Garg, sir, and also you to be to giving me the opportunity to present the lecture. So we can start now. Am I audible? Thank you, sir. I'm sharing my PPT. Then we will start. Presentation is visible. Saurabh, sir.
Yes, sir, presentation is visible. You are audible. Slides are moving. You can start, sir.
Okay. So today we will be discussing about the molecular docking and Autodock analysis. So first of all, we should know that this lecture is based on the computer-aided drug designing. So this is the latest thing which is coming to the medicinal chemistry, that on the basis of computational techniques, molecular designing can be done, docking analysis can be done, and various softwares are available in market, and pharmaceutical industries are also using these softwares to predict the hit or estimate the activity of the ligands or hit compounds. So first of all, we will see the introduction, and we'll be starting from molecular modeling. So what is molecular modeling? Molecular modeling stimulates the three-dimensional structural interaction between the atoms and molecules. That means we'll be creating the 3D structures of the drug molecule, whose activity we have to see, and the target receptor, the structure of the target receptor, or the protein molecule in which the drug has to be binded. So molecular modeling technique simulates the 3D structure of both ligand and the receptor molecule in which the drug has to do the binding. So we don't have to do the clinical trials of all the drugs. We can first of all go through the softwares to predict the activity of the drug molecules. So molecular docking can predict the optimized orientation of the ligand on its target. It can predict different binding modes of ligand in groove of target molecule. इसका मतलब ये है कि हम किस किस तरीके से ड्रग मॉलिक्यूल अपने टारगेट पे बाइंड कर सकता है. हम डिफरेंट ओरिएंटेशंस, डिफरेंट 3D स्ट्रक्चर्स के फॉर्म में ये विजुलाइज कर सकते हैं कि किस तरीके से बॉडी के अंदर रिसेप्टर पे जाके ड्रग बाइंड कर रहा होगा और उनमें से बेस्ट कॉन्फर्मेशन कौन सा है जिसमें बेटर ड्रग की एक्टिविटी मिल सके. So these methods have been used in the fields of computational chemistry. That means we are using softwares through computers, and we can do the drug designing. We can optimize the structure of the drug. We can do the computational biology, material sciences to study the molecular systems, and so on. So we will be doing this docking through a software called as Autodock. It's a very popular software, and in industries also, the research scientists are using this software to analyze the biological activity of the drug molecule. So Autodock is an automated suite of protein ligand docking tool. So as we know that the all the receptors are proteinaceous in nature inside the biological system. All the receptors or enzymes are proteinaceous in nature. All the drugs which are showing the activity inside the body, or the drugs which are showing the activity inside the body, they have to bind with the receptor, which is a protein compound, or enzyme, which is also a protein. So Autodock tool will predict the binding efficiency of the drug molecule with the receptor. So Autodock tools can be called as ADT, and they are designed to predict the protein interaction with small molecules such as drug molecules and substrate. The application of this tool is immense, ranging from structure-based drug design. That means on the by designing a drug, we don't have to synthesize the drug in the laboratory, and after that we don't have to do the clinical trials. But theoretically synthesizing the drug on papers, and we can know the structure of the drug, and knowing the structure of the drug, we can create the 3D structure of the drug through softwares called as CamDraw, Mervin Sketch. We can create the 3D structure of the drug, 3D structure of the protein molecule, and do the docking analysis. So protein docking can be done. Lead optimization can be done. Lead optimization means if we are having a drug molecule, what all functional groups can be added or deleted from the drug molecule to make it more active, more biologically active. And we can also predict the mechanism of action of the drug. We can also predict the structural activity relation of the drug with the help of this software. So large number of things can be done with the help of this software, and various versions of this software are also available, like Autodock 4, Autodock Vina. Some of the features are different in various two versions. So here we will be discussing about Autodock 4 version. So Autodock 4 is the software which analyzes the interaction of the drug molecule at specified target site of the protein. So as we know that the protein is a big molecule. Large number of amino acid chain combines to form a protein molecule. And what is the and our drug molecule is a small structure. It's not a big molecule, not a very big molecule. So protein is a big molecule. So we have to decide the target area in the protein where the drug can bind perfectly to show its action. So for that we have the grid box option in this Autodock software, which will predict the active binding area in the protein where the drug can bind perfectly. So I will be discussing step by step how we can install this software, and after that how we can predict the biological activity of that compound. So coming to the installation of the Autodock software, we can download this software from Autodock website and set up the installation files. Run the files named mgt tools. Mgt tools will be file will be coming, and we have to install that file. And after finishing the installation, we will be have the Autodock software in our system. So to successfully run the Autodock software, the protein and ligand structures in their .pdb format have to be present within the MG2 tool folder. So both the protein structure and the ligand structure. So receptor means protein and ligand means our drug molecule. Both of them have to be in .pdb format. That means protein data bank format that can be created through Autodock software. First of all, we have to prepare the protein molecule. So preparing the protein molecule is important. First of all, we have to get the protein molecule. So there is a website called RCSB. Here, all the proteinaceous structure or receptor structure of the receptors, structure of all the proteins, targets, what all diseases have been developed for that, what all targets can be, what all possible targets are there. The structure, 3D structure of all those proteins are available in the website called as RCSB. From this website, we can download the structure of protein first of all in .pdb format. Already I have told you, and we have to prepare the protein molecule for docking interaction. So here we will be removing the non-amino acid residues like water molecules, ions, ligands. These all molecules have to we have to clear. We have to get the clean structure of the protein. Not anything like water molecule or ions or ligand should be present in the structure of the protein. So we can prepare the protein molecule. For that, we have a different software called as Pymol software, or WordPad can also be used. So we'll be cleaning the protein molecule, and after that, execution of Autodock can be done. So Autodock software can be executed after preparing the protein molecule. So Autodock software, what will this software will do? Autodock software will calculate and predict the interaction between the ligand and the protein molecule based on the predefined parameters. So, on the basis of the concepts of chemistry, the parameters have been decided that how the ligand molecule will bind with the protein molecule. What are hydrogen bonds or hydrophobic interactions will be done, bonding will be done to show the biological activity of the compound. So, to be precise, the interaction between the molecules will be calculated at the specified region in the protein. So specified region means in the protein means protein is a big molecule. We have to select the area in the or the active region in the protein molecule where the drug molecule will be binding or will show the better interaction. So a grid map option is there to select the region in the protein molecule where the binding of the ligand with the amino acid will take place. Now, setting the grid box at the binding site or the active site or other essential regions of the protein is important because as we are seeing that the ligand have to bind with the protein molecule or the receptor for giving its biological activity. So, active region, that means the setting a grid box is one of the most important parameter for predicting the activity of the drug molecule. So, in Autodock analysis, we'll be taking the four major steps to predict the biological activity of the compound. So first step will be the initializing of the molecule. Second will be the running autogrid, running Autodock, analyzing interaction energy. Step by step, we'll be seeing all these parameters, discussing all these parameters. First of all, initialization of the molecule. In initialization of the molecule, we have two molecules. One is the protein molecule and the other one is ligand. So in initialization step, this will include the addition of hydrogen atoms and addition of Coleman charge to the protein molecule. This we will be doing in, we have to open the Autodock software, and we have to import the protein structure of protein, 3D structure of protein in Autodock, and the 3D structure of ligand. And first of all, we have to add the Coleman charges and hydrogen atoms to the protein structure. And while for the ligand molecule, we have to add the Gestiger charges for identifying the aromatic carbon, detecting the rotatable bonds, And to protein, the protein has to be initialized manually, while the ligand is automatically initialized when opened in the tool. So we have opened the Autodock software after installation, and we have imported the 3D structure of protein and the 3D structure of ligand. Now executing the auto grid. So auto grid is a active region. That means we have to select the active region in the structure of protein where the best bonding interaction of ligand can take place with protein. So auto grid has been executed to define the region and area in the protein to be analyzed for the interaction with the ligand molecule. After that, Autodock will analyze the interaction of ligand molecule and the amino acid that are present in the within the grid box. So setting up the grid box is a crucial step. So we want to see the interaction of drug molecule with protein. So for that, the setting of grid auto grid option is very important, because that is the region where the binding of ligand molecule will take place with protein. After executing the auto grid, we will execute the Autodock. Now, finally, we have protein molecule, ligand molecule, the active site of the protein. We have predicted the active site of the protein, that is the through autogrid. And when autogrid has been executed, after that we can further move on to executing the Autodock. So this is the final step, executing the Autodock. After that, we'll be getting the parameters which will show the biological activity of the compound. So what all parameters will be analyzed after executing the Autodock? We will see that further on. Once autogrid is successfully completed, Autodock can be executed. So Autodock calculates the interaction of the amino acid within the grid box and ligand molecule. So within the grid box, what all amino acids are present will interact with the ligand molecule, drug molecule, and the binding interaction can be predicted. Now, interaction has taken place. Protein, that means the receptor, and the drug molecule, the bonding or docking means binding of both the receptor and drug molecule has taken place. And on the basis of that, we will analyze the interaction energy, how both of these are interacting. Once Autodock 4 has been successfully executed, the result will be given as the 10 best conformations. These can be viewed in the analyze options. So after docking has been performed, we'll get the 10 best conformations. 10 best conformations means the structure of the drug molecule will be, the orientation of the drug molecule will be different in the 10 conformations, and the binding interaction, we'll see the binding interaction in out of the best 10 conformation, what is the best conformation in which the drug is binding with the protein molecule. So we'll having the 10 conformations. Out of that, we'll see which is the best conformation in which the drug is binding with the protein molecules. So this conformation can be viewed by play rank by energy. There is an option called play rank by energy. Through that, we can see the 10 conformations. The number of hydrogen bonds can also be visualized. One thing is that the drug molecule also forms hydrogen bond with the receptor molecule, and hydrogen bond is very important for the action of the drug. The drug which is forming more hydrogen bonds with the drug molecule, that drug shows best activity. Some drugs forms two hydrogen bonds, some forms three or four hydrogen bonds. So this is the Autodock panel showing the calculated docking parameters. After the docking has been performed, we will see a window like this in which the different parameters are showing. The first one is binding energy, how drug is binding with the protein molecule. So binding energy is always in negative. Here we are getting 8.21 value for binding energy. Ligand efficiency, inhibition constant, how much the drug is inhibiting the enzyme or a receptor. So inhibition constant value is 955 here, and its unit is nanomolar. So, and at last we are saying two hydrogen bonds are formed. So in this for this molecule, two hydrogen bonds are forming with the uh, receptor molecule. Okay, so we will get the parameter, these all these parameters after docking, and how these parameters will help to predict the biological activity of the drug. We can see this later on in next slides. So docking studies. In docking studies, I will take an example of a drug molecule. We are watching some oxazole derivatives, which will be acting on aromatase enzyme. So aromatase enzyme is a enzyme which is responsible for breast cancer in women. The production of this enzyme causes breast cancer, and there is a proven molecule, oxazole molecule, which shows better inhibition of this enzyme to for the treatment of breast cancer. So I will show you the five derivatives of oxazole and their binding energy, inhibition constant value after docking with the aromatase enzyme. So binding of protein with its inhibitor can analyze the molecular docking technique, can be analyzed by molecular docking technique. And after docking, we will be getting two values: binding energy, that is delta GB value, and inhibition constant, that is TI value. And I'm showing you one table which will be representing the docking parameters of compounds. In compound code, I have taken five oxazole derivatives, oxa 1 to 5. And here we have got the binding energy in kilocalorie per mole, inhibition constant in micromolar and nanomolar concentration. Here, binding energy is in negative, are the negative values. Uh, what all values we can call that it is a best value for binding? So, if the negative value is higher value from all these five binding energy values for all the five compounds, the negative value is highest is minus 6.88. This is the highest value. So that means the binding energy for oxa 4 is best. In case of inhibition constant value, which can come in micromolar or nanomolar concentration, as we have seen in the window. Uh, So here, the five values which you have got after docking, the minimum concentration value is 9.12 micromolar concentration. So the minimum inhibitory concentration, all these concentration out of the five concentration, this is 9.12 is the minimum concentration of oxa 4. So binding energy, this is best in oxa 4. Inhibitory concentration, minimum inhibitory concentration, that means in minimum concentration it is showing the inhibition of that aromatase enzyme. So by watching the docking values of binding energy and inhibition constant, we got that oxa 4 is the best compound which is binding with the aromatase enzyme, that means inhibiting that aromatase enzyme. So out of the five compound, this oxa 4 is showing the best results. So after docking, we will get the results like this in the form of binding energy. That means how the drug is binding with the aromatase enzyme target to its target, and how it is inhibiting that target. Now the interacting residues of the protein with ligand. Now we can also see what all functional groups of the drug molecules are binding with the amino acid residues of the protein molecule or any enzyme. So through figures, through 2D and 3D structures, I can show how all these compounds, oxa 1 to 5 compounds, are binding with the protein molecule. So molecular modeling analysis results are shown in table 2, and we can also see the 3D structure of 2D and 3D structures of binding of ligand with the protein molecule. So this is a table summary of protein residues interacting with the ligands. So as I have taken the example of five oxazole derivatives, and here these are the ligand molecule are the oxazole derivatives and drug molecules, and these drug molecules are forming hydrogen bond interaction and hydrophobic interaction with the protein molecule. So these are the codings. I have this comes in the form of coding, like V370 is valine. Valine is an amino acid. So In the aromatase enzyme, valine 370 amino acid is interacting with oxazole 1, and hydrophobic interaction, phenylalanine 430, 430, 203, 307. These are the codes of amino acids. That means in our aromatase enzyme, there's a long chain of amino acids, and this number shows which amino acid is interacting with the drug molecule. So in all cases, oxa 1 to oxa 5, hydrogen, oxa 1, oxa 3, oxa 4 and 5 are forming hydrogen bondings. That means these compounds will show better interaction with the aromatase enzyme. That is our target. And hydrophobic interaction, a lot of amino acids are also showing the hydrophobic interaction. Based on this interaction, we can also predict the structural activity relationship of the compound. In next slide, I am showing that docking interaction of different compounds with protein or enzyme. That means here I have shown the docking interaction of oxazole with aromatase enzyme. So this is the 2D structure, two-dimensional structure, and besides that, it is 3D structure. So in 2D structure, we can see that this is the structure of the drug molecule, and here the dotted line, green dotted lines are showing the hydrogen bonds. That means this oxazole ring, in this oxazole ring, oxygen is forming a hydrogen bond with valine 370. That means this is an amino acid. These circles are showing the amino acid, these dotted lines are showing the bonds. Green dotted lines are showing the hydrogen bonds, and purple dotted lines are showing the hydrophobic interaction. And on the basis of this 2D structure only, we have shown the shown the amino acid interaction with the drug molecule. Now we'll see the 3D structure of the drug molecule. This is our drug molecule in the pocket of aromatase enzyme. How it is, we can see how it is bonding with the protein structure aromatase enzyme and inhibiting this enzyme, which is responsible for cancer in this case. So these are the 2D and 3D docking interaction of the oxazole 1 with aromatase enzyme, and like this only, further we can see the docking interaction, 2D and 3D docking interaction of oxazole 2 with aromatase enzyme. In each case, we can see different amino acids are interacting with different functional groups of the drug molecule. So in all, we can predict how the structure of the drug molecule can affect the biological activity of any drug. How the drug will bind with the receptors inside the body. We can do this through docking technique and predict the activity of the compound. We can also do the drug designing through this technique. We can do the structural, we can predict the structural activity relationship of the drug compounds. So it is a very useful tool to analyze the activity of the drug in its target. So these are the docking interaction of all the 5 oxazole derivatives in aromatase enzyme, which is a target. So in conclusion, we can say that the biological activity of different synthesized compound can be estimated through molecular docking technique, and these methods have been widely used in industries to predict the hit or the potent medicinal compounds. It is also used to screen large libraries if we are preparing a large number of compounds and we don't want to go for clinical trials. First of all, we want that we don't want to invest in clinical trials in vivo process for predicting the activity. So we can go through software techniques to estimate or predict the activity of the compound. Further, if we find the hit compound or the active compound, we can go for its synthesis for its clinical trials. So besides docking, Autodock tools can also predict the editing the protein and ligand and modify its structure. Molecular docking is also a kind of computer-aided drug designing technique, and it can design the drug in various process through various processes. Like it can predict the binding and binding mode of already known ligands. Lot of drugs have been synthesized earlier, but no, docking have not been done. So for the already existing compounds, we can predict the binding mode, how they are binding with the receptor. We can also identify the novel potent drug compounds or ligands, and we can also predict the binding affinity of drug with the protein molecule. So this is one technique which is latest in medicinal chemistry, and we can go for this technique to predict the activity of the compound. So thank you.
Thank you, sir. It was really a wonderful talk on docking. You had touched all the aspects of docking, and nowadays, pharmaceutical chemistry is one branch where all the researchers are trying to do certain kind of practices which leads to innovative molecules. On behalf of ISF College of Pharmacy, Moga Punjab, once again, I am thankful to you, sir. You have taken, you are really, on a very short span of time, you have taken time from your busy schedule. So on behalf of Internal Quality Assurance Cell, I, Saurabh Kose, thank you once again, sir. And in future also, I will be in your requirement of your pharmaceutical expertise and research. Thank you, sir.
Thank you so much, sir, for giving me opportunity, and I will be always available if you will invite me for further lectures. Thank you so much.
Thank you, sir.